Welcome. My name is Christian Alejandro, and I will be presenting on the history of the ABO system in a medical setting, its molecular biology, and the evolutionary perspective and explanation that has been made hitherto. Whether or not we know what ours is, we all have a blood type. In fact, there are about 43 different blood types we all express, but the one you are most familiar with is the ABO system. This is because the ABO system has historical and contemporary medical significance in blood transfusions. Blood transfusions were understood well enough to routinely perform them for hundreds of years, but while the first successfully recorded transfusion was performed on a dog in 1665, blood transfusions were cited as having mortality rates of 33 to 40% even in 1901. Thankfully, someone by the name of Karl Landsteiner finally uncovered one of the mechanisms responsible for patient mortality in blood transfusions, which triggered a high volume of histological and biomolecular research which endures to this day. You see, there are two main things that go wrong in a blood transfusion with an incorrect donor-recipient cross-matching. One, the isoagglutination or clumping of red blood cells, similar to when you scrape your knee on something and it scabs up, except in the case of a transfusion, these scabs form within your blood vessels, clogs them, and can cause many complications such as arrhythmia, heart attack, or a stroke. The second possibility is a more confrontational immune response, where the complement system and leukocytes respond to foreign blood units by rupturing them open, and this is known as hemolysis. The consequence of mass hemolysis in the blood is the concentration of hemoglobin in the urine, which is also associated with a disease called acute tubular necrosis. Basically, your kidneys get starved of oxygen because all the free hemoglobin in the post-hemolysis serum sequesters it, and this can cause kidney failure. So what is the underlying mechanism for these immune responses? The immune response of the incorrect cross-match is a consequence of simple molecular biology. Each ABO blood type is the expression of a distinct surface glycoprotein. The most important part of the ABO glycoprotein are the carbohydrate chains at their ends. The template for the ABO carbohydrate chain is a galactose, N-acetylglucosamine, fucose, and galactose. This is also known as the O blood type. The other blood types A and B either attach an N-acetylgalactosamine or a galactose molecule to the chain respectively. Each blood type creates IgM and IgG antibodies to recognize glycoproteins that are not their own. O makes antibodies for both A and B makes antibodies for A, and etc. So the IgM antibodies are responsible for the isoagglutination response, while the IgG antibodies are responsible for the complement system and passive immune response. But why would we adapt to attack our blood if we didn't perform transfusions until recently? Well, there is no decisive answer yet, but for starters, it is certainly not because our ancestors were keen to performing blood transfusions. What we do know is that the ABO polymorphisms have been heavily conserved for at least 21 million years. To paint a picture, ABO polymorphisms are just as highly conserved as MHC class molecules used by the adaptive immune system and polymorphisms that produce tricolor vision. It has been discovered in at least 17 other primate species, including Old World monkeys, New World monkeys, and our closest ape relatives. The truth is, ABO glycoprotein antigens are expressed in more than just red blood cells. They are expressed in the saliva, vascular endothelium, mucosa in the gut, and respiratory and digestive epithelium. Most species outside of the great apes do not even express ABO in their blood cells, but more on that in a bit. The prevailing theory for why we have such highly conserved blood types is because they do appear to provide some sort of resistance to pathogens, and specifically gut-related ones. Modern research has shown that some blood types are in fact more susceptible to diseases such as gastric cancer, which is also a disease associated to gut pathogens. Coevolutionary conditions have been observed for pathogens such as the Norwalk virus and the Helicobacter genus, which have shown to adapt to ABO phenotype population frequencies. Most interestingly, the Helicobacter species have been associated with regulating a gene in mice that switches ABO expression from gut tissue and mucosa to blood. This has provided an avenue for researchers to pursue which can explain the connection between our blood types and its evolution from exclusive tissue expression found in more primal ape species to modern apes expressing ABO in their blood. This presentation was surely not as comprehensive as the research available on the topic, and I highly encourage you to visit these references for yourself and read them because it is quite fascinating that there's a bigger story to our blood typing than just transfusions.